Morning students. Today I want to discuss you about the major experiments related to photosynthesis in our 10th class biological science textbook. In first lesson there are four important experiments are there. Before going to deal experiments, I would like to share you how to write an experiment. In each experiment, it includes five major steps. The step 1 is aim. Aim means what you want to know from the experiment or what you want to prove from that experiment is the aim. Step 2, required apparatus or material. What the things you have to collect to do the experiment. These are the required apparatus or material for our experiment. Step 3, procedure. The way how you have to conduct the experiment. Step 4, inference. What the things you learnt or observed while doing the experiment or after doing the experiment, what you have learnt from that experiment is the inference. Step 5 is precautions. What care you have to take while doing the experiment. These are the 5 important steps to follow while doing or writing the experiment. Now we will discuss the experiment number 1 in our 10th class first lesson nutrition. The experiment is oxygen liberated during photosynthesis. We will start our experiment with the procedure what I have explained before you by stepwise. Step 1, aim. To prove that oxygen is liberated during photosynthesis. Our aim is to prove that oxygen is liberated during the photosynthesis. Step 2, required materials. For this experiment, we require beaker, test tube, funnel, water and hydrilla twigs. Beaker, test tube, funnel, water, hydrilla twigs. Step 3, procedure. Take a beaker with 3/4 filled water. Collect hydrilla twigs and insert these hydrilla twigs into the funnel. Put the funnel reverse in the beaker water. Then take a test tube and fill it with water. And completely filled test tube will be inverted over the funnel stem. The water level should remain in the test tube. We have to look after this before going to proceed next step. Place the arrangement in sunlight for 3 to 4 hours. After that, we will observe the gas bubbles coming from hydrilla twigs. They replace the air in the test tube. The water level in the test tube will be a little bit decreased and the air level will be increased in the test tube. Then we have to take out the test tube from the funnel carefully by closing its mouth with our thumb, because the air inside the test tube would be remained in the itself. So we have to close its mouth with our thumb. Then test the gas by inserting an incense stick into it. It will burn with more flame. Inference, the 4th step. More burning of flame indicates the presence of oxygen. So our aim is fulfilled. Precautions. Step 5. Ensure the level of water in the beaker above the level of the water in the funnel stem. So the level of water in the funnel stem will be low and the beaker water will be some high. Carefully take out the test tube from the beaker and close its mouth with thumb. These are the precautions for this experiment. So our experiment, oxygen liberated during photosynthesis will be fulfilled by doing like this. Thank you.